We're next going to go into part of the lecture series on cells and talk about cellular energy, the use of energy by the cell, how the cell makes usable energy, and also how the cells, certain cells, photosynthetic cells, create chemical energy, stored chemical energy from inorganic matter and inorganic energy, meaning sunlight. We'll start with cellular respiration because all eukaryotic cells conduct these reactions to generate ATP, and so therefore it's the most ubiquitous form of energy usage. All right, so when we were talking about the working cell, we indicated that the cells use energy for various processes, including the function of enzymes, which either put molecules together or break them apart into smaller pieces. The energy required by most of those processes is in the form of ATP, adenosine triphosphate, and it's been called essentially the molecular unit of energy currency within the eukaryotic cell. It is composed of essentially an adenine, sugar, and nitrogenous base, and then, three phosphate groups linked together, and the energy is stored in the covalent bonds that bond the phosphate groups together. So, cellular respiration is the process whereby the cell breaks down larger molecules. They can be proteins, fats, or sugars, and converts the energy in those chemical bonds to ATP, available energy to do cellular work. We focus on glucose in describing this process of cellular respiration because glucose is, one, the form that we use. We take glucose off of glycogen and feed it into our cells, and so glucose is the most important molecule from which we get our energy. But in addition, glucose is also the form in which energy enters the biosphere. It is photosynthetic organisms that take CO2 and water and solar energy and rearrange those bonds into glucose. So the focus on glucose is obvious. However, do keep in mind that we can also put proteins and fats through this process with slightly different chemical reactions and still gain ATP from those. So recall as we go through all of this that the energy and matter of life can be described as a chemical reaction going in either direction. Energy and matter come into our system through photosynthesis. Green things are the most important things on the planet at the moment as far as supporting the biosphere. So the components that are required to keep those green things growing are also highly important. Those components are CO2, a harmless, odorless, and colorless gas, and water. We are on a water planet. You take the energy from the photons of sunlight through the process of photosynthesis and those photosynthetic cells, and these bonds are rearranged into glucose, which is now stored chemical energy and stored biological matter. So this is now organic. So you take two molecules that are inorganic, an energy source that is inorganic, and life converts those into stored chemical energy and matter, organic energy, organic matter. And the waste product of photosynthesis is oxygen. Then what our cells do, we can't photosynthesize, but we can take the glucose that we get from eating the plants and take that molecule in the presence of oxygen and feed it through another system to change the molecules that are stored in glucose not the molecules, I should say, the chemical bonds, the energy stored in the chemical bonds of glucose into energy that's now stored in ATP, which is immediately usable energy. The waste products of that reaction are CO2 and water. So matter can go back and forth through these equations. Energy flows one way. It comes from the sun, is stored in glucose, goes to ATP, and then we lose it as heat into the system, and so we depend on more sunlight coming in to build the system up again. All right, cellular respiration is conducted for us largely in the mitochondrion. Mitochondria, as it says at the top of this slide, is plural. Mitochondrion, which is I-O-N, is singular. The mitochondrion is an ancient bacterial cell that was able to take the waste products of the original old eukaryotic cell and use them to get more energy. So the original eukaryotic cell could feed the waste it produced to this bacterium, and the bacterium could convert that into an immense amount of ATP. That generated a symbiotic relationship between the two types of cells to the point where eventually the mitochondria was incorporated into the larger cell and can now no longer live independently from the eukaryotic cell. And the energy provided by the presence of the mitochondria allowed the eukaryotic cell to become much more complex, larger, and so now the eukaryotic cells simply can't survive without the mitochondria and the ATP that is made available by that organelle. Mitochondria replicate themselves. They have their own naked loop of DNA like a bacteria does, like a prokaryote does, and they reproduce themselves on their own. They are typically inherited in the female egg or ovule if you're a plant. And we don't really need to go into why that is the case, but it is. There are 3 steps in cellular respiration. I want you to know the 3 steps, and in a sense, I want you to know where the energy in each of these steps. I go over that more in the following lecture on respiration. The 3 steps are glycolysis, which means the lysis, the breaking of glucose, the breaking of sugar. That happens in the cytoplasm of the mitochondrion. Then the products of glycolysis are put into the mitochondrion through a series of chemical processes called the Krebs cycle, and all of the carbon that was in the glucose, the 6 carbons that are in glucose, are converted back to CO2. So the carbon has now re-entered the inorganic world after the Krebs cycle is finished. And there's not a lot of energy that's usable created at that point, but the products of the Krebs cycle are then used in an electron transport system to build a cellular battery. And then that battery is allowed to run through this enzyme called ATP synthase, and that's where ATP is synthesized, that's where ATP is made. And again, that's within the mitochondria. All right, so here are 3 steps in sort of diagrammatic form. In the cytoplasm, glucose is split apart, so it's lysed. It's broken into 2 smaller carbon molecules. So it goes from a 6-carbon molecule of glucose to 2 3-carbon molecules of pyruvate. And there's a little bit of ATP yield. Then that pyruvate is moved into the mitochondrion, and through a process that leads to the Krebs cycle. Through the Krebs cycle, all of the carbon is lost as CO2, and you get a little more ATP. And then through the use of high-energy electrons attached to the products of the Krebs cycle and one of the products of glycolysis, you build the battery, and that battery runs through ATP synthase, and you get the lion's share of ATP through this oxidative phosphorylation using the electrons. So this is glycolysis in much more detailed chemical form. You will not be tested on all of this, but it is just to show you that it is a complex process, a linear process. Glucose uses 2 ATPs to get split apart into the 2 3-carbon sugars. Then each one of those produces an NADH, which is an alphabet soup molecule that will provide some of the high energy electrons later in the process in the building of the battery inside the mitochondrion. And then each of those 3-carbon molecules yields 2 ATPs on the way to becoming pyruvate. And it's pyruvate that is moved into the mitochondrion that generates the other alphabet soup molecules and high energy electrons to fit into the building of the battery. So we use 2 ATP, we gain 4 ATP, so that's a net of 2 ATP from glycolysis, and we also get 2 of these NADH molecules that we will come back to later. The pyruvate moves into the mitochondrion, and there's a process where 1 CO2 is kicked off of each molecule, and then the 2-carbon molecule ends up being attached onto a molecule called acetyl-CoA, coenzyme A. This gets actually pumped into the Krebs cycle, and again, there's a number of steps here which you will not be tested on, but you do get an ATP from each of the pyruvates that came in, so the Krebs cycle yields another 2 ATP, and so you've got a total of 4 ATP from glycolysis and the Krebs cycle, and all of your carbon is gone as CO2 by the end of the Krebs cycle. What you do have more of, though, is these NADH molecules with high energy electrons, NADH, NADH, and FADH2 with high energy electrons, and then more NADH. So from passing through the membrane of the mitochondrion through the Krebs cycle, each pyruvate yields 4 NADHs, so that's 8 for each glucose molecule, and 1 FADH2, so that's 2 for every glucose molecule. So at the end of the Krebs cycle, you've got 4 ATP, 8 NADHs, and 2 FADH2s. And now your energy is largely in the electrons of those alphabet soup molecules, NADH and FADH2. All right. So then NADH and FADH2 feed their electrons, the high energy electrons, into a series of enzymes that are embedded in the inner membrane of the mitochondrion. Remember, the mitochondrion has 2 levels of membrane. There's an outer membrane and an inner membrane with lots of folds in it that create a lot of surface area on which these reactions can take place. Electrons are donated at the beginning of the enzyme chain by NADH, which yields a hydrogen ion and NAD, ion. The electrons pass through the enzyme system and back out into the matrix of the mitochondrion, and they attach to oxygen. So then oxygen becomes negatively charged on the inside matrix of the mitochondrion. The energy from the electrons passing through the enzyme system is used to grab protons, hydrogen ions, and pump them across this membrane into the space between the outer membrane and the inner mitochondrial membrane, the space called the intermembrane space, meaning between membrane space. FADH2 feeds its electrons a little bit further down the chain. They're not quite as high energy, but that yields a couple more hydrogen ions, and it pumps a few more hydrogen ions into the intermembrane space. So the mitochondrion is building up these negatively charged oxygens here in the matrix and these positive protons here in the intermembrane space. So you've got a positive pole, a negative pole. Positive wants to meet negative. That's the running of a cellular battery. The protons are allowed to re-enter the matrix and join up with oxygen, making H2O, water. And as they do that, they cause this enzyme called ATP synthase to crank down and snap a free phosphate onto an adenosine diphosphate. It's got 2 phosphates and not 3. You add this third phosphate by the action of this enzyme, and now you've got ATP. So the mitochondrion is regenerating ATP by attaching the ADP and the free phosphate to ATP synthase, and then having the proton movement through it crank and snap those two pieces together into ATP. So it's not a direct chemical reaction of the chemical bonds of glucose breaking and transferring directly their energy to ATP. It's an indirect reaction through this building of a cellular battery. All right. Now, the respiration we've talked about is called aerobic because it uses oxygen. The yield from this reaction is essentially the 4 ATP through glycolysis in the Krebs cycle, and then 28 to 32 more ATP from the electron transport system and oxidative phosphorylation through ATP synthase. So anywhere from 32 to 36 ATP produced from each glucose with the presence of oxygen. Oxygen is required to pull the electrons out of that enzyme system. Let's go back to that slide real quick. Oxygen has to be here to grab the electrons out of that system. They're very low energy, and you need a highly corrosive, highly reactive substance to pull them out of there at such a low energy state, and that's where oxygen comes in. Oxygen is able to do that for us. That's why if you don't have oxygen, you suffocate. You basically get the electrons clogged up in the system and all your mitochondria, and so you can't produce ATP. Without the vast amount of ATP that gets generated by this oxidative phosphorylation and ATP synthase, our cells simply can't survive. Now, the two forms of respiration that don't use oxygen are called anaerobic respiration. One is fermentation. This is what yeast does when it's essentially making beer, or if you're using yeast to make your bread rise, it's creating ethanol and carbon dioxide as a waste. And another form of fermentation is lactic acid fermentation, which is what you do in your tissues if you don't have oxygen available. So if you're doing some heavy exercise, your body will quickly start burning through your glucose and use up the oxygen that's there. If you're not able to replace that oxygen through breathing and distribution of that oxygen through your bloodstream to those cells in a rapid manner, then your body will switch over to lactic acid fermentation in those tissues, and you'll get a couple of ATP out of each glucose. Eventually, you'll feel fatigue, you'll have to stop, and then your breathing allows your cells to catch up with the oxygen they didn't have. You feed oxygen back into those cells. The product of lactic acid fermentation goes backwards, and you convert it back into pyruvate, and then it's able to go through the entire Krebs cycle and oxidative phosphorylation on the inner membrane of the mitochondrion, and you get all the ATP. You just have to catch up on your oxygen supply in order to get all of that. Okay, in the next lecture, we'll look again at where the energy is and where the matter is as we go from glucose to ATP energy.